Okay, this training video is going to be a quick introduction um, to DNA and translation and transcription. Very rough, very quick, but just to make sure everyone's on the same page. So, very quickly, so DNA. Actually, I have to get that on the screen. Hold on. Move myself to a corner. All right. So, DNA. Let's look at what DNA actually looks like. So DNA, as you know, is the backbone of genetics. Um, so it basically looks like this. So this is the actual chemical structure of DNA. And so DNA consists of 4 different genetic letters. And basically, there's 4 structures that govern DNA. 4 different types of DNA molecules, and those are adenine, thymine, guanine, and cytosine. Okay, so you can see here that basically the backbone of the of DNA is the same for all of them. Okay, so this backbone, and what the difference is the base that's attached varies. All right, so these bases now. Honestly, knowing the chemistry of this is not incredibly important when doing just general general genomic studies. That's actually what makes bioinformatics possible, because bioinformatics doesn't store details about chemistry; it stores details about which particular bases are in sequence. So there's a so the ends um, of the sorry the direction of DNA is based on the connection to the to this sugar here, and so there's a 5 prime end and a 3 prime end. Okay, and so DNA is usually read from 5 prime to 3 prime. Okay, so you'll see that's how they mark out um, the parts uh, of this sugar. So anyway, the thing about DNA is that so we've got 3 letters. So DNA, that is an awful D, sorry. It's got 3 letters. It's got G, C, T, and A. Okay, so these guys pair with each other. So G always pairs with C, C always pairs with G, T always pairs with A, and A always pairs with T. So if I've got a DNA strand, so DNA always is stored in chromosomes in pairs. So if I have a DNA sequence, and I'll just make up a DNA sequence, A, C, G, G, T, A, T, C, G. All right, so if this is the 5 prime end and this is the 3 prime end, so this is a DNA sequence, then on the other strand, you're going to have so A always pairs with T, C always pairs with G, G always pairs with C, G always pairs with C, T pairs with A pairs with T, T pairs with A, C pairs with G, G pairs with C. Okay. Now on the other strand, you're going to have the opposite. This is the 5 prime, and this is the 3 prime. Okay. So it goes this way on this strand, and it goes this way on that strand. And so when the DNA is being replicated, replication only occurs 5 prime to 3 prime. So replication is always 5 prime to 3 prime. So what happens when it makes this strand, which is known as so let's say that DNA is going along this strand. Okay, what it does is it splits this guy into two segments, and on the leading strand, it just synthesizes away. Okay, it uses DNA polymerase to make copies of this guy, and it's happy because it's going 5' to 3'. But if you think about it, this strand is going the opposite direction. So what it does is on the lagging strand, it will synthesize short segments going this way. So a lot of individual segments so it's going left to right, but little bits as it goes left to right, it's doing the other one right to left in little bits. Okay, and then it's joining these together using a DNA ligase. So that's the way that replication works. So on DNA, there's replication, and that's how it makes copies of itself. And then the way that it makes proteins is through two processes. I mean, you could split it up into a number of different processes, but the two main ones are transcription and translation. Okay, so transcription is where you have DNA, and so again, I'll just make up a sequence. C, G, A, T, A, C, G, G, C. Okay, so this is 5', this is 3'. So what happens in transcription is that, so basically what happens here is in transcription, we make messenger RNA. All right, so RNA is used for a lot of the intermediate products. DNA gets transcribed into messenger RNA. Okay, so then this messenger RNA then gets translated into proteins. Okay, so and it uses what's called transfer RNA to make that happen. So basically goes DNA to mRNA and then to proteins, and that uses tRNA to do that. Okay, so we're going to go through the process. So if you take a single strand of DNA, transcription works on a single strand. So before you get to what you're translating, there's a whole bunch of stuff that happens beforehand. There's a transcription start site, and it has that's basically where a bunch of molecules attach, and it basically opens up the DNA and says, here you go, you can start transcribing here. There's a lot that's still unknown about this process, but one thing that often happens, a lot of things in genetics often is often and not always, because we don't know all the details. Often there is a what's called the TATA box, and it's usually TATAAA. And so this is upstream from where transcription starts, but it kind of aligns all the promoters so that they're going in the right direction. So it knows, okay, well, we've opened it up here. This kind of shows that the direction is going this way, and it gives a good clue both to the molecule and also to us reading the DNA that this is going to be some sort of a coding gene. So then you have kind of a leader region that has kind of stuff that gets transcribed to mRNA but doesn't necessarily get translated to proteins. And then you have a start codon, and we'll talk about that in a minute. And then it starts copying, and then you have the actual DNA that's going to get generated into proteins. So let's say that we have all the header stuff, and we're moving along. What happens is mRNA will synthesize a corresponding mRNA transcript according to what's in the DNA. So if this is our DNA, the mRNA will get complementary pairs. All right, so these are, now instead of thymine, you get uracil. So that's the one change. So other than that, it's the same basic bases, and you just get a complementary strand of mRNA. And so this then gets sent out, and there's a few modifications on it. It gets a cap and a tail, to stabilize it. And so the cap, I believe, is a guanine, and the tail is a poly A tail. So anyway, so that kind of stabilizes it in the cytoplasm, prevents it from being broken down. So this can then be spliced. So there's a lot of alternative splicing mechanisms. So DNA can make more than a single protein, and so alternative splicing can take a single messenger RNA, and depending on the needs of the organism, can resplice that to different transcripts. So it gets processed and spliced, and then there's actually structures called spliceosomes which do the splicing. Sometimes the RNA can actually splice itself. The parts that get removed are called introns, and the parts that stays are called exons. Okay, so introns are removed and exons stay. All right. So then we've got this mRNA transcript. What that's going to do is it's going to be then translated into proteins. And so what you're going to do is you're going to have tRNA, which is going to be complemented to our mRNA, and it's going to do sequences of 3, and it's going to translate these into amino acids. So every 3 base pairs gets an amino acid. So for example, so there's going to be, let's go to my DNA table to figure out what these translate into. So I've got it's wrong page. So here is, we'll go from the DNA table. So we have, what do you start out with? GCU. Okay, so what does that translate? Or actually, the DNA is CGA. So what does that translate into? Well, you go to this table. The first base is C, the second base is G, and the third base is A. So that's going to be arginine. Okay, so there's going to be a transfer RNA molecule that is going to be also be CGA, and it's going to have an arginine attached. Okay, well then we're going to have TAC. All right, what does that go to? We'll go to our codon table. T, go over to A, and so TAC, that's tyrosine. Okay, so going to give us a tyrosine. It's going to be attached to a tRNA that is it's here. No, this, yeah, UAC. That's going to be a tyrosine. And then this one, GGC. So G, G, C, so that's glycine. Okay. So that's going to be G, G, C. And so that you can see that the mRNA, which is this one, the mRNA pairs with the DNA in a complementary way to produce the mRNA strand. The mRNA then in the ribosome gets attached, has little transfer RNAs come along and bring the amino acids with them, and then these bond together to form a protein. All right, and so this table is kind of the standard DNA table. Now, some organisms have slightly different DNA tables, but they're all basically the same. So now there's also a couple interesting things. ATG is oftentimes used as the start codon. So especially in eukaryotes, almost all transcriptions start with an ATG for the beginning of the actual gene. So there's a leader region that doesn't get translated, but ATG tells it where to start where to start actually translating. Then you have 3 stop codons. All right, so these tell it where to stop. And so all of these have meanings. You can see that more than one DNA code translates into these different amino acids. All right, so and some of them have more flexibility than others. So you can see there's a whole lot of them that translate to leucine, but there's only one of them that translate into methionine. Okay, there's only 2 that translate into cysteine. Okay, and there's a lot of reasons for this. I think only some of them have even been uncovered, but it allows the genetic code to actually be used in really interesting ways. So this is known as a degenerate code because there's more than one source for every destination. But in addition, so one reason that's been stated is that it allows for some amount of mutation without affecting the actual protein sequence. And while that may be true, I think a more interesting reason may be that there is meta information encoded in some of these different alternative versions. It allows you to have a code within a code. So you're kind of embedding meta information about the structure itself. So kind of it might be used to influence splicing, might be used to target mutations. It can be used for all sorts of things. So anyway, that's the codon table. That's how to use it. There's different representations of the codon table. Let's see, I thought this had another one, but maybe not. There's a biocomplexity paper that called a tetrahedral representation. So there's oftentimes a circular one, but what they propose is that we can actually convert this to a tetrahedron that has the start codon at the apex and the stop codons on the other 3 corners. So anyway, there's a lot of ways to represent this. The easiest one for lookups, I think, is this standard code, this standard table. So anyway, so that's how it works. That's the RNA table. This is the DNA table. I think that's what we were working off of. So that's the genetic code. That's how it works. Next we'll talk, and very little of the actual chemistry is needed for understanding a lot of the action of these things. So this is why we often store it as letters. and we can refer to it as letters without hardly any loss of understanding or relevance, because that really is operates as a symbolic code. Um, and so a lot of the machinery of the DNA and RNA are there to keep it as a symbolic code. So anyway, so that's the basics. So we've got replication is when DNA makes copies of itself, and so just actually there's a article. Oh no, it's translation. So replication is when DNA makes copies of itself, and again, it copies the leading strand directly. The lagging strand, it does little segments. This article from Nature I thought was interesting. It has kind of the basics that we talked about, but it also has a little bit more details, a few things that we found more recently. So this is a good little intro to it. Um, so so translation occurs in multiple steps. First, you transcribe the DNA into RNA. And so this is mRNA. The mRNA goes to the ribosome, and the tRNA attaches the amino acids, which makes the polypeptide or protein. So, so that's the basics of DNA and how it works, and that's it for this video.